Hi, my name is Obineche, and I'm majoring in mechanical engineering at Hostos Community College.
And I am Scarlet Martínez Cardoze, majoring in chemical engineering at the City College of New York. Together, we are working with our mentor, Dr. Yoel Rodríguez, on computational biophysics. Specifically, we are performing in silico discovery of neutralizing agents targeting SARS-CoV-2 spike glycoprotein. The SARS-CoV-2, also known as Severe Acute Respiratory Syndrome Coronavirus 2, was first discovered in December of 2019 in Wuhan, China, producing the COVID-19 pandemic. This virus causes a large number of symptoms affecting the respiratory system in particular. The spike glycoprotein plays an important role in virus infection because it recognizes the receptor HAC2 and facilitates the viral entrance into target cells depending on the binding of the surface unit. This coronavirus currently has 5 variants. The original one was produced in Wuhan, China. Then in the United Kingdom, South Africa, Brazil, and India were discovered the other 4 variants. In this figure, we show the 3-dimensional model of the SARS-CoV-2 membrane and the spike glycoprotein that are attaching to the membrane in the outside. In this figure, HAC2 starts interacting with the spike glycoprotein, inducing conformational changes. The spike protein ended up penetrating the healthy cell via its membrane. And because the spike protein is so important for the virus replication, our objective is to find a small molecule hit compounds with high affinity and selectivity for SARS-CoV-2 spike protein receptor binding domain or RBD. We hypothesize that if we do find these small molecules with high affinity and selectivity, this could prevent the interaction between the HAC2 and the spike protein RBD and therefore affect the SARS-CoV-2 replication.
The research was done in 2 different steps. The first step involved the structural analysis of SARS-CoV-2 spike glycoprotein in complex with HACE2, PDB-ID7DF4. 3D models were built based on the available structure, PDB-ID7DF4, for the 4 different variants at the time this research started: the D61G, the South Africa, the UK, and the Brazil variants. Visual inspection of these models was conducted to identify the main interaction between the SARS-CoV-2 spike glycoprotein receptor binding domain and HACE2. The second part of the research involved the structure-based feature screening of the e-molecule database. About 4.3 million small molecules were bombarded into the spike protein receptor binding domain to determine the molecules with the highest affinity to disrupt the interaction between the SARS-CoV-2 spike glycoprotein and HACE2. Visual inspection of these molecules will be performed by first ranking them using the Sigma-4 scoring function. Thus, approximately 50 molecules with the highest affinity to disrupt the interaction will be selected for further experimental testing. For the preliminary results, the first diagram shows the zoom portion of the interaction between SARS-CoV-2 spike glycoprotein and HACE2. The box on the left side shows the HACE2 interacting with the SARS-CoV-2 spike glycoprotein. The residues highlighted below are the main ones involved in the interaction between the spike protein and HACE2. The second diagram shows the grid box built in blue are the spike protein receptor binding domain and HACE2 protein-protein interaction to conduct structure-based molecular docking virtual screening.
As it can be seen in this table, 10,000 small molecules for each model out of the 4.3 millions were selected using the CheMOS4 scoring function and ranging between -10.8 and -6.29 kcal per mole, including both UK and SA models. As an example, here the main interactions of the small molecules selected for the UK and SA variants and the CheMOS4 scoring function energy decomposition distributions are shown. In addition, visual inspection of these small molecules was conducted to select the top-ranked compounds to be tested experimentally using the SARS-CoV-2 spike pseudotyped lentivirus assays. In conclusion, we were able to identify the SARS-CoV-2 spike glycoprotein main residues involving its interaction with HACE2. The computational methodology seems reliable to predict SARS-CoV-2 spike glycoprotein small molecule inhibitors, and about 50 top-ranked compounds have been advanced to test experimentally. For future plans, the best binders will be chemically optimized to be used as lead compounds. Thank you all for listening.